All right, hello everybody. I want to welcome everybody for coming here tonight to the Koch Institute for our celebration of the FDA approval of Lumicell. My name is Matt Vanderheiden. I'm the director here at the Koch Institute, and this is part of our year-long celebration of our 50 years of cancer research at MIT. And I can't think of a better way to celebrate than to all come together and really hear the stories of what I think truly embodies what is possible when we bring people from different perspectives together to try to solve big problems in cancer research. The story today, the team that you will hear from, really came together to develop a bold idea. They dared to ask a question, and that question was, what if we could detect residual cancer cells, make surgery better, and avoid recurrence? Could this save lives, and could it push boundaries? I have to say, so some of you who know me know that I trained as a clinician, and I was actually taught in medical school that this was not possible, right? I was told that there was no reason to do something like this because we already knew what surgery could and could not do. But nonetheless, they didn't listen to my medical school professors and decided to pursue this line of research separately. And the outcome is a groundbreaking new advance for breast cancer patients that was recently FDA approved. Now, I'm also particularly proud of this because this is something that truly is homegrown here at the Koch Institute. Like many places, we need a way to fund early stage crazy ideas that aren't going to garner the traditional funding mechanisms. And for this purpose, we have the Frontier Research Program here, whose really mission is to seed bold new ideas, get them off the ground, and see where they might go. Now, this has long been one of our signature programs, and not everything turns out quite the way this did. However, this story is one that I think is really spectacular, because from a small seed investment many years ago, we now had formation of a company and a new therapy for patients, and I'm excited to have the people who were involved in that come together here and tell you about their story. So at this point, I'm going to turn it over to Tyler Jacks, the founding director of the Koch Institute for Integrative Cancer Research, and he will introduce the Lumicell team as well as be your MC and host for this evening. Thank you so much, Tyler.
Well, thanks very much, Matt, and again, welcome to everybody. This really is a wonderful occasion to celebrate. You'll notice at the top of the slide, this is actually the 50th anniversary of cancer research at MIT, and we've had a number of programs over the course of the year to celebrate that milestone. And this is our latest, and I think it's the last of the year, actually. And it's a wonderful way to bring this year to a close, to celebrate a true accomplishment of cancer research here at MIT and of the Koch Institute. The Lumicell experience embodies what we try to do here on so many levels. It represents the bringing together of individuals with experience in different domains to work collaboratively to tackle difficult problems and with some frequency make success, and this is an example of success. It's also an example, as Matt mentioned, the value of investment by our community in our investigators. Matt mentioned that we have had and continue to have a Frontier Research Program that provides funding for early stage ideas. Some of them will germinate and grow like this one did. Others will die on the vine, but that's okay. And these investments are critically important to advance our work and to stimulate the kind of cross-disciplinary research that we're going to hear about today. This particular program was funded by the generosity of Kurt and Kathy Marble, and Kurt Marble is sitting in the front row. I used to have the pleasure every summer of meeting with Kurt and Kathy to talk about the work that fund supported. And I remember very distinctly talking about this project in its earliest days, not making any promises that this would, in fact, bear fruit. but with the hope that it would. And as we'll discuss tonight, it clearly has. It's clearly changing surgical outcomes and has the great likelihood of changing survival and reducing the cancer burden directly. So that's a wonderful ending to this story. I have a personal connection to this story. Some of the research that led to it actually took place in my own laboratory, and Linda, I think, will emphasize that point. But in addition to that, shortly after I became the director, I was chatting with my neighbor on Otis Street in Newton, and that neighbor was David Lee, sitting in the front row. David had become a friend by then, but he had no real connection to cancer. He was working at Analog Devices at the time. He was an MIT grad, class of '69, master's in '70, so we had that bonding to share. And we talked about things that were developing in the Koch Institute at some frequency. But then tragedy struck. David's wife, Ramsey, was diagnosed with breast cancer, and it was, in fact, terminal. Ramsey lost her life to the disease. And so over that period of a couple of years, David completely changed his orientation and decided that he wanted to commit himself to making a difference in cancer research. And that became a singular focus for him. And we began to discuss how that might manifest itself. And he expressed an interest in getting more involved in the Koch, and we did that. And then he got engaged in discussions with lots of faculty here, including these two. And that led ultimately to this proposal that is now Lumicell. So I have a long connection to this project, and I'm extremely proud of the work that they and their team has done to bring it to this point. So we're going to run the program in the following way. You're going to hear from each of our three speakers, all of whom played a critical role in the development of the Lumicell story. David will start, and I've told you a little bit about his background. One thing I didn't mention already is that David is a lifelong learner. He's one of those, and they're not uncommon around here, these MIT undergraduates. They never stop learning. David loved to learn just about everything. But as an example of that, he got his master's in 1970, and he did all the work that I mentioned. And right now, he's doing his PhD in mechanical engineering, and he will defend his thesis in 10 days. With the formation of Lumicell, David took on leadership positions there, including CEO and CSO. He stepped back from those roles now and is a member of the board of directors, but obviously still very deeply involved. We'll next hear from Linda Griffith, who's a professor here in biological engineering. Linda is an expert in a range of fields, including tissue engineering, cell biology, various aspects of engineering as applied to the life sciences. She's interested in how tissues form and function and how they become dysfunctional in the context of disease, with a special emphasis on diseases of women, including cancers. And you'll hear a bit more about Linda's story. Munji Bwendi is a professor in the Department of Chemistry, an expert in nanotechnology and in particular nanocrystals, and was a discoverer of quantum dots. If you don't already know what quantum dots are, you should realize that you watch them on your television probably every night. Munji has been an expert in that area and its applications in many domains, including biomedicine. And he was awarded the Nobel Prize in 2023 in chemistry for those observations. So we have a star-studded lineup to tell you about the Lumicell experience. And once they've finished their discussion, I'll invite them up to the front of the room, and we'll have a further conversation about where Lumicell is and where it's headed, and really what they've learned from this process. So thank you for being here. We're looking forward to a wonderful evening, and I'll have David begin. Thank you.
Well, while he gets that ready, I'll tell you the rest of the prequel. I met Tyler and talked about my desire to be involved in cancer in the driveway between the two houses. And, you know, that was an interesting setting. He was not incredulous and opened the door to me to come to the Koch and participate. And it was a wonderful experience. He also was the one that introduced me to Linda and Munji. And without that introduction, it wouldn't have been a team. And he was also the person that obviously gave us the funding to get us started. Without Tyler, none of this would have happened. So, see your neighbor in the driveway. It can be very... So I'll start with kind of the background. You may know these things, but the origin of this is that cancer resection is difficult, as Matt said. Between 9 and 36% of cancer surgeries leave cancer behind, so there's a second surgery. That range is because there's different techniques that surgeons use. Our experience is much closer to 20 to 30%. What is important there is that of those re-excisions, about 65% of those don't find residual cancer, so tissue's taken, but there was no cancer removed. And very importantly, about 14% of the time, the pathology misses the cancer, the patient is sent home, and there is residual cancer remaining. And that is one of the things that we tackled. Just to give you a kind of a brief idea of what the technology and how it's used is there is an injectable, this is a drug, and we developed that. That was, just as a side note, I think we did that in 6 months or something. But getting it through the FDA and demonstrating that we can produce it in volume, that was one of the things I'll mention that was unexpected. That took years and tens of millions of dollars. The development was short. The proof that you could take it commercial was a long time. The lumpectomy is removed after the injection. The handheld device then detects the cancer. It does detect it to microscopic levels. I think in one of our pictures you'll see that there's a very small cancer that was detected early on, and we have a picture of that. The remaining cancer is removed. You check again to make sure that you got everything, and that's it. So as this one is kind of, I like this one because it kind of tells the story of where I was coming from. When I was talking to Tyler, I was at Analog Devices making MEMS devices, and I said rather naively, the things that I work on are about the same size as a cell. Maybe I can do something in cancer. And, you know, that is funny, but it turns out that's exactly what happened. As I looked at it, I realized that pixels in the cameras that we were making are about the same size as a cancer cell. And therefore, we could use a simple low magnification camera to look at the cancer cells. So that was the idea, that you just have pixels that can see the cancer cell, and it's a kind of a simple, straightforward mechanical engineering approach. We had to make a drug. This was, as I said, took some time. This was Linda and Munji and Jorge, who I might be here, developed this drug. What you can see is that it gives you the boundary between tumor and healthy tissue. The brown is the Lumicell 15 lighting up the tumor, and it is very effective, very sensitive, and is at the heart of the technology. Not easy to do. I think Linda will give you a few words about what had to go into that. In terms of clinical impact, I might say a side note here. This presentation is a little modified from what we did back in 2017, and this is kind of a side note, because back in 2017, we had the freedom to say pretty much anything about performance and our future. Having been approved at the FDA, we can't do that anymore. So any statements that we make, like this one, has been approved by the FDA. And as a result, I can't talk about the trials, but you can find them on the trial.gov, and you can see the things that we are doing. Just type in Lumicell. But what I can tell you is that there was a significant reduction in second surgeries. There was improved outcomes. The number that I think is, to me, significant here is that the residual cancer that we detected that was removed, that was missed by the standard of care. So these are the patients that would have had, that had surgery, pathology would not or did not detect any residual cancer, and they would have been sent home. And the fundamentals are that the lump is taken out, pathology takes about 2% sample of the tumor and extrapolates about what is being left behind by looking at the margin. And all the cancer isn't contiguous to the margin is one problem, but they're also only looking at a very small fraction of the tumor. Whereas, as you saw, we are scanning the entire cavity and we are not extrapolating about what's left behind. We are seeing what's left behind. And so it's very effective in getting all the cancer. This is the team back in, it had to be before 2017. I think everybody still, in my opinion, looks the same. But then, well, maybe. As you can see by the list of capabilities and competencies, it is, just as Tyler said, cross-functional. Ralph Weissleiter is at the bottom in the bottom left. He is at MGH. He helped us with the drug development. David Kirsch was Tyler's postdoc, who at the time we were doing this had gone down to be the faculty at Duke. And he's where he conducted the early stage work. That was the preclinical, namely testing everything out in mice, and then the phase 1 clinical trial. I'll mention something about the phase 1 clinical trial, and I think Linda and Munji might remember this. The drug we made was an all-new chemical entity. It had never gone in a human. And I remember the night before the first injection, and I was thrilled that we had the FDA, because if you tried to do something like that on your own, without the coverage of and protection by the FDA, it wouldn't have happened. And it went well. There was no problem, but first in human is a big step. A lot of trials do not involve first in human drugs, and it's difficult to conduct that trial, and it was done at by a special group down at Duke. And as I said, at the beginning, this was all Tyler. And the early funding from the Frontier and the Marble Cancer. These are the things that made this possible. And with that, I'm going to go to Linda. I miss you.
Thesis defense.
Oh dear, no questions though, right?
Clear. Okay, yeah. All right, so It's really, really fun to be here tonight. And I want to underscore something very strongly that Tyler said about David always wanting to learn. He called me when he thought about going back for his PhD around Christmas time, whatever number of years ago that was, and he's like, do you think I can? I was like, I know the head of the admissions committee. I'm calling him right now. And the head of the mechanical engineering admissions committee had also just lost his wife due to metastatic breast cancer. So the MIT is like this community that understands that people are going to make progress in helping the problems that are really intractable. So at the time that we started, that Tyler put us together. You know, David comes to MIT and he goes around talking to people. Where is the, how do I go forward? Do not remove. How do I make it go forward? There we, oh, okay, sorry about that. So at the time, the thing about David and being so willing to learn is we would get together in the office, Munji's office, with usually some chocolate or snacks on Friday afternoons, and no question was off the table for being ignorant, okay? We were very different backgrounds, and I found that David's biology was very, very spotty, and my optics even spottier, okay? However, Munji and I were teaching physical chemistry together, so at least we had some common knowledge about a few of the processes that may contribute to making a device and drug that might work. Okay, so at the time, I had started then in the middle of my career working on a disease called endometriosis, which has many features similar to cancer. It's an invasive disease, it's inflammatory, a lot of immune cells, and we were very, very interested in the protease biology of these diseases. And we're in fact inventing some tools to study the biology in the lab dish. Now, it's a lot different to study biology in a lab culture dish than it is to put something in a patient. But when we got together, you know, David's asking, well, can we use surely there's an antibody. I'm like, well, there's all kind of tumors. And, you know, sometimes a tumor, even if it's got the ligand for Herceptin, not all the cells in the tumor will express it. So that probably isn't a great approach. The other things, people were really excited around that time about using ligands for cancer cells, like folate receptors should be folate, not foliate. And again, that was very cell specific. And when you think about cancer biology, the tumors are really heterogeneous. They've got all kind of expression patterns across the tumor. There's a lot going on. The cell that's migrating, we had a lot of discussions about catching the cells that are migrating out that may have very different biology than the cells that are in the main part of the tumor. So it's like, well, what if we just did something that's pretty non-specific, but may be shared by a whole bunch of all the cells in the tumor, or in the microenvironment, there's just so much noise and drama going on from a particular function. What if we did that as a way to look at the tumor boundary, something non-specific that would just catch everything, okay? So that's what we ended up doing, targeting both the tumor and the immune cells. Anybody causing havoc in the general vicinity would be there. So then we're saying, okay, now how are we going to do this? So the drug, you know, it's one thing again to have something like we were using in lab, works in a culture dish and you image it with your microscope. But the drug in the patient had to be really, really different. First of all, it had to accumulate where the tumor is, okay, in the margins of the tumor. So we had a lot of discussion about the physiology of tumors. And there's something called the EPR, where you have leakiness of tumor vessels, a lot of debate about that. And so you don't want it to bind randomly to a lot of other parts of the body. And this is where some of the drugs come in. It has to be activated by proteases that are very prominent in the tumor boundary. And those, at the time, I was looking at mainly MMPs, but cathepsins are extremely strongly expressed across many of the cell types. You have to be able to see it deeply enough to know to go in and scrape out the extra tumor if it's there. So it's got to be bright. And you want it in the patient's tumor margin for a long enough period of time at the level of brightness against the background, because you got to have signal against any endogenous fluorescence in the tissue. It's got to be bright enough for a long enough time that you can inject it into the patient in some setting where she can have injections before surgery, and then she goes to surgery, and the surgery lasts about an hour, and you need for it to be bright in a particular window of time that's relevant for the patient. So we had to have all of these kind of things. We had many, many discussions about all of the different little features and designed many, many different compounds. And David Kirsch was really incredibly helpful, as was Ralph Weisleiter, in helping us look at these in animal models. And we learned, for example, from David Kirsch that cats get cancer when you inject them with vaccines. They're prone to cancers at the injection site. So we learned a lot of things about the potential models and came up with the, okay, I'm really bad at this. Okay, so we came up with the idea, which was not incredibly novel idea, but getting it to work properly in the tumor setting was really hard. And this is to use what's called a quenched fluorophore, so that you've got a little peptide that can be cleaved by the proteases in the tumor microenvironment, a lot of them there. And when it's so there's two molecules attached to this peptide, and one of them will be fluorescent when you hit it with a certain wavelength of light, but when it emits at its fluorescent wavelength, the quencher absorbs that. So if it's wandering around in plasma completely intact, it doesn't shine, doesn't fluoresce. But once you cleave that, the quencher's gone. Now that fluorescent molecule can be very bright. It turns out, we found out that the cells actually could take this up inside, in the tumor margin, could accumulate some of this fluorescence, which was good. We didn't love it that they get brighter and brighter and brighter as time goes on. So this was the heart of what we did, and that gave us the kind of brightness that we needed. And there was a lot of debate about which, you got to have a particular fluorophore that can go into patients. It can't just go into the cell culture, it has to go into patients. And so there was a lot of factors that went into choosing the particular fluorophore. We also wanted to have it be bright, again, against the tumor tissue. So in animal study, so this is the molecule that we came up with, peptide with the quenchers, and enzyme acts on it, and it becomes, there we go, that's not, yeah, and then it becomes fluorescent. So that's basically how it works. And it accumulates in the tumor. So now we have the drug, and with meeting all the requirements. And what was really, really incredible about this is you could see it in large tumors. You could see the margins showing up on the top, the images, and then the pathology image. But you could also pick up something as small as ductal carcinoma in situ. So little tiny tumors. And so patients with a large central tumor may have other little foci of tumors, because the tumor cells, particularly certain types like HER2 positive, can stick their fingers out and go wiggle around. And if you have a large tumor, the pathologists, like we'll hear later, I got breast cancer right after we started working together, and I had a 3-centimeter tumor, and they only did 23 slides of a 3-centimeter tumor. So who knows how many of the margin cells could have snuck around that Lumicell would catch, because you catch every single one. You scan the whole cavity, you catch everything, and they're really bright. And I loved David's slide about the pixels, because you can see little teeny tiny bright. And some of the tumor margin work that we showed, that David showed, Shannon Alford's name was on there. She was a postdoc actually with my husband, Doug Laufenberger, who's actually part of the Koch, and she's now a program manager at NCI, and she manages systems biology programs at the National Cancer Institute. So this had a huge impact, this program had a huge impact on the way she thinks about her career in translation. Did I catch everything? I can't. Okay.
A little green light here. Should be on. So I should be on. There we go. Okay. I first met David Lee when he was program manager for a cancer nanotechnology excellence NIH program, or at least he was part of that program that was led by Ralph Weisleiter and Bob Langer. And that's when I first met him, when we were brainstorming together. I think you were involved in this as well, Tyler. We were brainstorming together, and I had no idea who David was at the time, but he was the guy in the room. And then there was a workshop that was put together by the Koch on cancer biology. And I didn't know very much about cancer, but so I went to it because I wanted to learn more. And David was there at the same time. And then at the coffee time, we really started talking to each other. And he asked me some questions, which, like, could you see a single cancer cell in vivo? And my background was in looking at single molecules, in doing microscopes, single quantum dots, and of course you can see a single cell. That's so much bigger than a single molecule. Even when you label a cell, you're labeling it with thousands and thousands of fluorophores, so that's like drop dead easy to see in a microscope. And so we began talking through that, and as Linda mentioned already, David is not afraid of asking any question that he's curious about, and I found that so refreshing. And then the first idea was actually, could we look at cell signaling? Could we invent some sort of stick that you could go into a tumor and then label different parts with different colors, and then look in real time at the signaling events. And so we spent many days thinking about how to engineer that. You're looking at new avalanche photodiode detectors that might be sensitive enough to do that. And then I think it was David that had a conversation with David Koch, where David Koch said, my unmet need is I really want to be seeing leftover cancer in situ. And so David Lee came back and said, this is the unmet need. And I thought, boy, this is easy to do optically. And I told David, this is easy to do if we want to do it, but the hard part is the biology. Because you've got to be able to specifically label the cells. Once you label them, you can see them. And that's where Ralph Weisslater came into play, because there was this little company that was supposed to be doing just that. And it turned out that wasn't completely true. So we spent a lot of time designing the instrument, thinking that somebody else was going to take care of the labeling, the hard part, which was the biology. And then at some point, it turned out, well, the biology stuff really wasn't worked out. And so that's when we had to design our own drug from scratch. And we used all of our expertise. I had been working with John Frangioni first at Beth Israel, then Rakesh Jain at MGH. So I had a fair amount of experience about the kinetics, the biokinetics of drugs and the clearance through the kidneys or through the liver from my work with quantum dots. And then Linda came in and explained to us all about biology, which I really was lacking, even more than David probably. I was so happy when Linda would ask, or when David would ask questions, and Linda would, start explaining. And I think at one point I turned to David and I said, I'm so glad you asked that question because I have no idea what the answer was. So it was a great time. I think we all learned a lot. And at the end, we had this molecule that, and we specifically chose, Cy5 as an agent, because for a couple of reasons. One of them is because it had been used in a lot of clinical trials before. And so there was a track record. And from what I had learned before is that you want to be very conservative with the FDA. You don't want to come in with something that's really completely brand new, because you're just asking for trouble. And so having technology that wasn't completely new, this was fairly old technology, and the drug, the fluorophore was also not completely new. And the second part that was, in retrospect, quite good was that Cy5, at least the agent that we were getting, was about to go off patent. And so the cost of that drug was about to plummet, which turns out to be really important. The cost, because in the end, cost is so important. And anytime you want to bring something to market, it can't be expensive, right? And so we had to design a handheld imaging system that was inexpensive, that you could almost give away. And then the drug itself needed to be, $1,000. It couldn't be, and that includes profits. So the cost of goods needed to be quite low. And so that was also part of the design, really important. And, so in any case, the conventional thinking at the time was that we needed to go into the infrared, where penetration was deeper, and we needed to have sub-millimeter resolution to see inside the cell, and that a lot of the systems were big systems with microscopes that look down onto the patient, and they look straight down, but it turns out that when you're doing surgery, the patient isn't flat. It's a three-dimensional object, and you're taking a lumpectomy, there's stuff on the sides here, and if you have something that looks down, you're going to completely miss that, right? And so, thinking about the optics of it, if we're going to be looking for the margin, that means you're looking for cancer cells that are at the edge, not very deep inside. So, a few millimeters inside. So if you're exciting with light that's reasonably blue, the penetration depth is actually not very deep. It's just deep enough to catch those cells, and then it gets absorbed by that part, and so you're then not contaminated by extraneous fluorescence that's further down. So the choice of color then becomes important. It limits your depth of interrogation, which is what you want. The red light, we don't need to go infrared because, again, we're looking just at the surface of the, within the few millimeters, and the red light can come out. And and this is another thing that I learned from working with the clinicians before coming to Lumicell, especially with Frangione at Beth Israel. You have to work within the workflow of the surgeon. Yeah, you can't add half an hour or 45 minutes to a surgery. That will never fly, right? So you have to have something that's fast and that's easy. And so if you're going to have sub-millimeter resolution, that means that, and you've got a couple centimeter field of surgery, that means you have to scan. And scanning takes time. And so you need to be able to take a picture in one shot. You have to be able to take a 1 centimeter by 1 centimeter or 1 inch by 1 inch picture in one shot. And so it's like doing astronomy. When we look at stars, we're not interested in the structure of the stars, usually. We're interested in, is it there or is it not there? You look at a telescope, the stars are little points of light. So the cancer cells are like the stars. They're little points of light. And in a telescope, a low-resolution telescope, you have a huge field of view of the universe, right? And then you see a gazillion points of light. And that's what we needed to do here. And so in order to get the best signal to noise, as David pointed out, if you have the size of the image on your pixel, or one pixel, be the same size as the cell, basically a 1:1 or a 2:1 magnification, where the fluorescence from each cell is captured by one pixel, that gives you the best signal to noise. And who cares about what's inside the cell? Is it there or is it not there? And so with that kind of design, we chose red light, blue light for excitation, so that we have a few millimeters of penetration. White field of view, but on the order of 5 to 10 micron resolution, the size of the cell. And we worked within the cavity, which means that we had something that needed to be handheld and just didn't look down. It had to be small so that it could be manipulated by a surgeon. And with that, we were able to show that, in fact, you could label single cancer cells and place them, in the cavity and actually see them. And this is an image of single cells labeled with this Cy5 agent that you can see. In practice, the surgeons don't need to see single cells. You never need to be at that resolution, at that sensitivity. In practice, what we found is that when there's stuff left over, it's actually pretty big. It's on the order of millimeters even. That's hundreds of cells or even more. So there's plenty of light. There's plenty of signal. So this is an image of one of the first uses of the device here. I don't know if David is actually in the picture or not, but David went to a number of them, and Dave Strasfeld, who's in the audience, also went to a few of these surgeries. And by working hand in hand with the surgeons, Barbara Smith in that case, designing the probe to be so that it's easy to use. We had models of tumors and breast and stuff like this, and how should it be shaped? What's the angle that of the basically, or all the ergonomics was really important as well. So we could actually, surgeon could actually scan around and get everything. And so first it was the David Kirsch's first trial was actually on sarcoma. And that was where we learned a lot about the biodistribution and how much drug we would need to use. And then we went into the breast cancer field. And these are a few quotes from patients from one of the early clinical trials. And having the patients give you this feedback is really gratifying. I mean, this is why we do these things. First, we do it because we're curious, and then there's a challenge, and we try to meet that challenge. And at the end of the day, when you take that and you apply it, and you've got feedback, and the lack of side effect, very privileged and grateful to have been in the trial. The results in my case could have been much different had I not been. I think this is somebody where we found some residual cancer that hadn't been caught by pathology. I wish you and Lumicell team the very best of luck. It could very likely change the course of history in cancer diagnosis. How wonderful that would be. So, and since then there have been many, many other quotes that Lumicell has gathered. And I think that's the end of my presentation here, and thank you.
So I'm going to invite my friends to join me up here, and we'll have a discussion amongst ourselves, and I encourage them to engage each other in the discussion, which I think is more interesting for the three of you and also more interesting for the audience. And before we finish, we'll open it up for questions, so please be thinking of questions that you'd like to ask our panelists. Let me start with where you began. So I'll frame this by saying, microdevices engineer, tissue engineer, and a nanotechnologist walk into a bar. They really walk into a chemistry professor's office, and they start a process which results ultimately in the success that we just heard of. What's the secret sauce in your view? You all touched on this a little in your comments, but what makes this sort of thing work, where you bring people together from these disparate domains of knowledge around a problem? What's the secret sauce?
You're best to answer.
Okay. It was just a really fun group of people to tackle a problem. Everybody was curious, everybody knew different things, and nobody was proud. Everybody wanted to learn from other people, and everyone was motivated to do it for very strong reasons. Obviously, David was really the glue. He went around, when you brought him to MIT, he went around and just met people. He wanted to learn about what's biological engineering, because the Koch had gotten started, there was a lot, and he just wanted to learn, and he talked to people, and he's like, this is somebody, this is a team that can do things together.
But I want to add another thing that I wanted to mention is part of the secret is also the students and the postdocs that we have at MIT, because we talk together in the office and we discuss big concepts, and we can make this thing, and we can design this drug, etc. But at the end of the day, somebody has to do it, and it's not us. And so I had a graduate student, Lisa Marshall, who was doing single molecule spectroscopy in my lab. No background whatsoever in biology, but she could design optics, and so she's the one who actually designed the first optical setup. And then we used little spheres, fluorescent spheres, to test it out. And then we hired Jorge, who wanted to be part of a group that could potentially spin out a company. And he had a background in fluorescent microscopy, and he also then followed Lisa Marshall. And then we hired Dave Strasfeld, who was a postdoc in my lab, then switched over. Again, no background in biology, but a strong background in optics and a desire to learn and to be part of the group. And so that the community of students and postdocs that we have here is really critical.
Yeah, I'm going to come to you in a second, Dave, but I just want to double down on this point. We're incredibly fortunate here at MIT. We've got super talented faculty, but we also attract the best and the brightest from all over the country and, importantly, all over the world, who are curious and motivated, who like to dig in and apply themselves to these kind of problems, and we couldn't do it without them. So what about you, the outsider? What was the contribution of the outsider's perspective here?
I guess I'd say the passion. I just wanted to do it. There was an interview of Munji and myself about this, early on, and somebody asked Munji, well, what did you think about this in the early days? And he said, well, as a faculty member, he said, well, he saw the risks. And he said, this had risks. And they asked me, and I said, well, I had no doubt. And, that's because I had no knowledge. But as you guys have said, a lot of interest, but it was passion. I wanted to do this. I walked out of my life as I was a director of engineering at Analog Devices, and I left that.
So, related to that point, we have a colleague here in the building, Bob Langer. Many of you know Bob. And he talks about his disappointment in applying for grants and getting rejected by all the experts who talked about the various reasons why an idea would never work. And Langer says, I had one advantage, which is I didn't know any of that stuff. And so that kind of naive perspective without getting burdened by what other people think will or won't work is critically important.
Yeah, there was, Munji reminded me, there was that moment when I asked, well, can we see a cell? And he said, oh, well, no problem. If we can do this, this, and this. And so that was the roadmap.
Yeah. So you all began this project back in, I'm going to say, 2006, 2007 timeframe. Kurt and I were thinking that the original funding was around then.
Yeah.
That was 17 years ago.
Oh, my God.
2017, you made a presentation, I think, in this room, talking about the Lumicell experience, and I think even then you were thinking you were on the cusp.
Yeah.
That was 7 years ago. So, simply put, what took so long?
Yeah. There was a part of it in the early stage where I wanted to keep when we formed the company, I wanted to keep the costs low, so we didn't build a big staff. There was just 3 of us. And for 5 years or so, all the way through phase 2, it was a very small group. And that was, nobody was complaining, and that was what I wanted to do. And then I think 2017, we were heading into the pivotal trial. Pivotal trial is the one where, phase 1 is safety, and in our case, safety. Phase 2 is efficacy. You got to show that it's going to work before you get approved to take it to the next one. The pivotal is phase 3, and that means that you've got to take it to many locations throughout the United States and have a lot of different players because it's supposed to simulate going to market. And I didn't appreciate what that would involve at the time. And the other thing that took time is that it's a drug and a device. And I am not absolutely sure, but I think there are only a few approved combination products in the last 10 years because you've got to get the drug approval as a full-up, brand-new drug, an NDA, and you have to get the device approval full-up on its own as from CDRH. And then it's got to work together, and you got to get the results that you have hypothesize that you're going to get. And that took more time than I thought. The other thing is that the company was looking at, as I said in 2017, I felt that we were 2 or 3 years away. And so the company needed to think about how to go commercial, because what you don't want to do is get approval and have nobody that can either make or sell your technology, and then you've got 2 or 3 years of, they call it the valley of death. And you didn't want to do that. What I didn't appreciate is the difficulty of getting a commercial team that was capable of going from where we were into the market. That, I thought, was, as an engineer, straightforward. But it's difficult to get the right combination of people with enough knowledge on both they've got to be able to transition from technology and also have a good sense of how to go to market. And that's difficult.
So now you've gotten to this point, and let me just point out that we invest in a lot of early stage ideas here. We support a lot of research. We attract other forms of funding to support research. It's not every day that we get an FDA approval of something that was homegrown. So that's why I'm saying this is a celebration. It's really very, very important. You guys have brought that to fruition. Munji, your last slide showed a bunch of quotes from patients. I find that incredibly impactful. So I'd like each of you to talk about the personal nature of this for you. You've now done something that is impacting the lives of cancer patients. What does that mean to you?
I've been talking.
Linda, you can start.
All right, I'll start, because right after we started working together, I got diagnosed with triple-negative breast cancer. And triple-negative, I found a tumor when I was adjusting my bra strap at a conference in Singapore, and it was a big one. And Barbara Smith, who's a fabulous breast cancer surgeon at the forefront of doing what's called breast conserving surgery, had just started to advise our project. And so I got in to see her forthwith. And you know, so she did the surgery, and then I had a big chemotherapy and so on. But during that process, it became very, very real what it meant to wait for your margins, because you have the surgery, you go home, and I was lucky it was node negative, so I got to go home that day, and I had to make a decision: do I have mastectomy or lumpectomy? And there was no difference in outcomes in the data, so I actually just had a huge surgery for endometriosis, so I wanted to not have another big surgery, so I did the lumpectomy. But we brought my billing records in to see, like we were trying, like, how much can we charge for this? I'll bring all my billing records in. And David went with me. I had the same medical oncologist that Ramsey had, and he came with me to my chemo, which was very meaningful to me, and talked to Dr. Weiner, who at the Dana-Farber. So it was actually both good and bad, because it really helped us focus on the patient experience and so on. And then again, last year, I got diagnosed with breast cancer again, and I had also not been that involved in the latest Lumicell, and I go see Dr. Smith, and I presume I'm just gonna have this technology, which I know is so much better. As I mentioned, there were only 23 slides for this 3-centimeter tumor, but I had massive chemo, and this time it was DCIS, early stage, but still a grade 3 tumor. And I'm like, oh, at least I'll get the Lumicell. So they'll go in and they'll cut any little things. It's DCIS, so I'm not getting chemo, but any little things. And she's like, no, it's at the FDA. It hasn't been approved yet. So, I'm still nervous about that last surgery because I have radiation, but it's very meaningful to pay. Like, I really, really wish I had been able to have that technology, because it's going to catch anything.
How about you, Munji?
Well, I didn't have breast cancer, but I had a different kind of cancers during the time that we were doing this, about 13 years ago now. And I went into the world of, being a patient, going through chemo, through the waiting times, through the the statistics, just, you worry so much, and it just, it overtakes your life completely. And, I'm very lucky. My outcome has been excellent, but it's not always the case. And anything you can do to improve the odds is massive at a personal level for the patient. And that's the most important part, but for society as well. Think of all the families that are impacted by cancer. It's just amazing. I spent so many sleepless nights thinking about my family during that time.
So David, we started talking about this in 2003, 2004. You were on a mission. It's maybe not mission complete, but it's a major milestone. So how does it feel to you?
It is important on so many levels. The obvious was the mission at the beginning is still compelling because I observed, I wasn't the patient, but I observed what my wife went through, and I resolved that there could be a better way. And I feel very pleased where we are. What I would say is that the other thing you asked, what does it mean to me? Besides meeting that goal, the other thing is that I guess it made me arrogant about, maybe that's not the right word, about being able to tackle difficult things. If you've got people like Munji and Linda and Strassfeld and Jorge, who are all the people that are here, and try something that is a reach. I'll just make a pitch for my next one is in neuroscience, and it's definitely a reach.
David told me earlier that he's going to solve the memory problem as we age. I'm all in favor of that. Linda.
One more comment that occurred to me as we were speaking, and I'll try to be short, is that Lumicell technology has the potential to help women make decisions that may make their lives easier, in that I counsel so many women who get breast cancer. I'm like the go-to person for anybody in the universe. Oh, can you talk to this friend of mine, whatever. And women are so scared. They want to have a mastectomy. And it's always, like, even my best friend would not have a lumpectomy because they're just, cut it off, cut them both off. And it's a big surgery, and there's a lot of complications you can have. And I've had 9 surgeries for endometriosis, so I just wanted the littlest surgery I could have. But the data for outcomes is what is so important. And as we move forward, I am confident that the Lumicell technology will help reduce recurrence in ways that will be meaningful. In ways, when I went in and had to decide mastectomy or lumpectomy, I looked at the data, and Dr. Smith had all the data, and it's like there's no difference in outcome. And so it's like, okay, well, I'm opting for little surgery, but having data that really, okay, now the outcomes are even better because you didn't have those 27 people who had the little rogue cells running across. Right. That, to me, will really help patients because so many people I know who've had mastectomies have had complications.
So let's finish up our conversation, then we'll open up to questions. But I want to finish on this point. Where are we headed? So you just mentioned, Linda, the outcomes. We talked about the approval based on changing the need for second visits and second surgeries. But there will be data one day to talk about outcomes. Are outcomes better? Recurrence rates different for people who got the Lumicell technology versus not? So I'm interested to know, to the extent that you can talk about it, where are we headed in that direction? And you began in breast cancer. Where else are you headed in terms of cancer indications for this technology?
I'd say a few things about that, if I may. Then, back in 2000, whenever, it was such a long time ago, the technologies that were available for the algorithms and for our analysis, were computer size. And now you can buy it on Amazon as a chip. I'm not being facetious. You can buy the kinds of processors that we would need for under $100 and run multispectral, more complex and definitely better and more sensitive algorithms. I'd like to see that as soon as we can. I know the company's thinking about that. So that is an opportunity that's right there to take advantage of, and people call that AI. I mean, I just call it good, solid algorithm processing, but the underpinning of it is taking advantage of what AI has done to processing, image processing. And then we are in trials in other cancers. And as I say, you can look at online. I've been told that I can't mention what they are for I guess, litigious reasons or something, but apparently there is an issue that once you've gotten FDA approval for an indication, what you don't want to be doing is indicating to people that it applies to other indications, because then it may be used off-label, and that's a problem. But we are in other trials.
So seeing the last cancer cell, you talked about you don't need to see every last cancer cell, but I could argue that you really would like to see every last cancer cell, because that last cancer cell might be the one that comes back and kills you. So do we need to push the technology further to increase our resolution and our specificity, in your view, or is where we are today good enough?
So I think the resolution is, we need to have resolution better than a cancer cell for the kinds of stuff that we're talking about. Sensitivity is always an issue. You can do that either with better biology, better targeting, and also with a combination of image processing. And so I think that's a direction that's definitely important to go into. Yeah, because they're, differentiating between different kinds of tissues, between different kinds of cells is not always easy. And so going to multiple colors and using more sophisticated software. And sometimes I think, medicine is kind of like NASA. The kind of computers that were on the space shuttle or on, up in satellites are decades old in terms of technology, right? Because it takes so long to get everything approved to be space ready and stuff like this, that you're always way behind on the technology than you'd really like to be. And I feel like it's the same thing here with these sorts of technologies, because the clinical trial process and then the FDA approval process, which took a whole year basically out of the hands of the company, during that time, technology just marches along at a rapid pace, and then you can't go back and upgrade along the way, because you're basically locked into a particular technology as you push through the trials and through the approval process. So there's always updating to be done.
I want to open it up for questions. I know we have microphones roaming around, and there they are. If you have a question, right here.
What are the other cancer that you are going to look into other than breast cancer? Are you going to move into throat cancer, pancreatic cancer?
So Jorge, you're going to answer this. I can say it? No, I'd rather you. So Jorge was my first hire, Strasfeld the second. Both Mungi's postdocs.
Hi, yes, I'm Jorge Ferreira. I'm now Chief Scientific Officer at Lumicell. I started as a postdoc at Mungi in 2008 when they convinced me to start this project with them. To address the question, and actually to clarify something like David said, we can talk about all the indications. What we have to be clear is that they're not approved by FDA yet. So we are looking into prostate, we're looking into brain, we're looking into esophageal cancer, gastrointestinal cancer, colorectal cancer. We're going to start looking into lung cancer also, and I think I'm probably missing one or two. But it's broad because the application is not just for breast cancer, it's solid tumors.
It might be worth emphasizing something that Linda mentioned, the drug in this case is detecting proteases that are commonly upregulated in cancers across different tissues and in immune cells across different tissues.
It's not particular. Any cancer, yeah, pretty much.
Yeah, so this.
Yeah.
To find something that was quite
And in the case of this, it's, you know, one of the slides said, you know, some people made the criticism, well, it's not specific, you know, infection or inflammation, other places in the body can have this. And in fact, you know, one could think about using it for the, but when you have a tumor, you are looking at a specific place. So if there's an infection in your toe, you know, if you're having boob cancer, you know, it's not going to be a problem.
Yeah, the concept that, as Munji said, the concept early on was, and Linda presented, if we went for ligands and binding, we were likely to have either just a single indication business, or we were going to have to make a drug for each indication.
And I'll add, so when I got breast cancer the first time, I was on the front page of the New York Times for having breast cancer because I had a situation where I was what's called ambiguously positive for the marker HER2, for which there's a great drug, Herceptin, and now all the antibody drug conjugates, really great drugs. But I had to decide whether to take Herceptin because I was weakly positive in part of my tumor by immunostaining, like right on the border, and then you do something called FISH, which is fluorescence in situ hybridization, and I was right on the border there too. So it's like, I'm not negative, but I'm not really positive, so in the case of having an agent that would target, you know, there's just so much heterogeneity. And so I was really convinced after that we had made the right decision.
Yeah, and as time went on, it became more evident because the protease and the inflammation that activates the drug is in the tumor margin, which is the mix of inflammatory cells as well as some wandering tumors, which is how it grows, and that's what we light up. You don't really need to light up the tumor. That's been taken out, and by the surgeon, it's sitting on the table. You really want the margin.
Question here.
Hi, great work. Although I'm a researcher, I'm asking this question as an outsider. What is the future, and especially how long it would take for this amazing technology to available in most of the hospital?
How long will it take to
Yeah.
Okay, David.
Well, it should start next quarter. It's, you know, there's production timing, and also we have to raise a lot of capital to make the drug and the device. And so I think those are the two pacing items.
Further questions? Yeah, in the third row there.
Oh, no, he can't ask a question.
Wait, this is less of a question and more of a comment. Tyler, you had asked what David Lee, as an outsider, brought to the group when they were first thinking about, you know, how to address this unmet need in cancer surgeries. And what I think got kind of glossed over is the fact that David is both a world-class mechanical engineer and a world-class product developer. And if you're an academic in this room who is interested in commercializing your technology, you want someone like David Lee in the room because not only are they going to contribute to conversations about what's possible, but also contribute to contribute their understanding around the details of how to get there. And so, you know, I suspect that when David first got started with Munji and Linda, neither of them knew what a Gantt chart was or what a quality management system is. They probably still don't. And, you know, David was kind of there because he had this deep-rooted experience in product development to kind of, guide them towards what commercialization might look like. And so, yeah, just to reiterate, if you're an academic who's thinking about commercializing some exciting technology that's being developed in your lab, having a David Lee in the room when you're having these initial conversations is incredibly invaluable.
That's a really great point, and I think it's something that we in academia could do a better job of. I think we only learn that lesson very slowly, and we don't put it into practice nearly enough. So thank you for bringing that up. There's a question back here, and then we'll have a question here, and then maybe we'll have some refreshments.
Thank you. I just have a comment, kind of a question, kind of a comment. I mean, on the one hand, it is great. You don't know what's not possible, but as David, as you said during your comments, did it stretch your timeline? Is there anything that you would have done? Yes, it's good to be naive. You don't know what's not possible. You push it, but what would you have changed to make it what would you have done differently to move things along a little bit faster? Would you have brought in other advisors? It also makes it a little bit tougher from a fundraising standpoint, because the people outside will see, well, you have a drug-device combination. Do we really want to, we know what kind of road is ahead.
Tough question. I guess, as I said, during the early years, 2008 to, I'm going to say 2015, I think the pace was something that I guess I personally set, and I'm responsible for that. My wife, Eve, called those my Camelot years. Working with the two guys, both of whom you've heard from, Ann Mungie and Linda, were, I woke up every morning just happy. And so I was resistive to changing the pace all the way up through the phase 2 trial. We could have raised more capital. There was venture capital to be taken in during that time. And I just did not go down that path. We actually wrote, I've forgotten how many, 5. We wrote 5 and won 5 NIH grants, for I think a sum total of $15 to $20 million total. That's how we moved. We could have accelerated that by a factor of 2, I'm sure. I'm absolutely sure. And then, as I mentioned, the transition between phase 2 efficacy and the pivotal, I think we lost 4 or 5 years. And that was because we didn't navigate the transition from a science team to a commercial team very well.
We'll take one last question up here.
I'll ask them after.
Yes.
First of all, not a question, a comment. Thank you so much for getting this approved, because I'm also a breast cancer patient, and I recall the days when my margins weren't clear and we had to go back in and do further surgery. So congratulations, fantastic news. And you've completed the kind of the scientific success. You've gotten the regulatory approvals, and I think now you're facing, okay, how do we make this a commercial success? It's great to get the scientific and regulatory approvals done, but now you have to deal with reimbursement agencies. You have to get on people's a list of approved device drug combinations, at least in the U.S., ex-U.S., you have to get the various government agencies to approve. I'm assuming you have plans in place for that, and then a roadmap for how to take this forward. You've named a couple of the other solid tumor indications that you're targeting. I'm assuming that's based on looking at where else this technology could go next, as well as where's the most commercially viable places to take it. So I'm just curious to hear a little bit more about those plans, because you can manufacture the drug, you can manufacture the device, but how do you get it into the hands of the docs?
Yep. Well, we were designated a breakthrough technology, which helped us. So we do have, as you mentioned, we have CPT code, we have codes. What that means is both the HCPCS and the CPT code. What that allows us to do, what that does is it takes away the burden of usage by any hospital or doctor because it's reimbursed. Now coverage, that's a second thing. Coverage means the efficacy has to be sufficient for the insurance companies to actually pay the money. And that's still ahead. But as I say, I think the major step is raising the next round of capital, which needs to be significant in order to go commercial. There are some strategics. These are large companies that are in the business that we might have a relationship with, and we've been looking at that.
I wish you luck with that too. I hope that next year we'll be sitting here and you'll be telling us about commercial success of launch.
Yep. Yes, thank you. I hope so too.
So on that hopeful note, we'll bring this part of the discussion to a close. There are refreshments outside, and we encourage you to hang around and ask our panelists even more questions. But I want to close by thanking them for their time and their efforts. This is a wonderful story of an idea born from a guy on a mission who was fortunate enough to meet two brilliant professors at MIT, build a successful team, and execute a plan. And let's not forget about the guy who provided the original money that got it off the ground. Let's thank them once again.